Morning. Thanks for coming to the Nerd Talk. Yeah, so my title's not quite as witty as yesterday, but hopefully it'll be fun. So I'm going to talk about improving gene drive, which if you were here yesterday, you should know all about gene drive, by knockout of non-homologous end joining in Aedes aegypti. And so Aedes aegypti is just mosquitoes. I'm also going to go very quickly through the introduction because yesterday was the introduction, and I went 20 minutes over when I practiced this morning, so I got to sprint. All right, so males are smaller than females. This is a male. These are the ones that suck blood and lay eggs. And so the reason that they suck blood is for the extra protein from the blood so that they can make the eggs. And so that's why only females suck blood. So. And then I had like this exact same slide yesterday. So the life cycle of the mosquito, the females suck blood. About 2 days later, they will lay eggs. And Aedes aegypti in particular will lay eggs just above the waterline. And so when it rains, the water level will increase, the eggs will get wet, and then they'll start hatching out. And so this is what some eggs looked like. I lined these up early on when I started working in the lab. It's not very straight. I've gotten a little bit better. And then this is the size of the eggs with my finger as a scale. And then these are larvae. There's 4 different stages of larvae. These are probably in this 3rd or 4th stage. And so these are lined up on a petri dish. And then these are pupa. So after their last molt, they'll be pupa. And so I will go more into why their eyes are different color. I teased that a little bit yesterday, but you'll know why their eyes are a different color today. All right, and so the range of Aedes aegypti in the US, you can see even Lubbock is still in the range of Aedes aegypti, and it's most of the southern US. The diseases spread specifically by Aedes aegypti are most of the viruses that we associate with being mosquito-borne. So yellow fever, dengue, West Nile, Zika, and chikungunya. And then some of the other diseases spread by different species. Malaria is mostly by species in the Anopheles genus. And then heartworms. Heartworms can be spread by Aedes aegypti, but there's some other species that spread heartworms also, but heartworms in dogs. So I talked about the control strategies in more detail yesterday, but just a quick review. There's mechanical control strategies, so that's ways of physically keeping mosquitoes away from people, like mosquito nets, or just clearing out water reservoirs that they can lay eggs in. There's chemical, which is insecticides. Biological, so there's different species of fish that will feed on mosquito larvae, and then I talked about some other bacteria that we can use as pesticides. And then there's genetic control, which is what I'll focus in on today, particularly CRISPR-Cas9, which is a gene editing technique. And so just to explain CRISPR-Cas9, so it's a site-specific way to edit genes. And so if you have a particular sequence of DNA here, then you can target that with the Cas9 enzyme. And the way that it's targeted is you have this hairpin of single guide RNA, or sgRNA, that has a sequence that will be complementary to that. And so that goes in, and then if it's just the guide RNA complex to the Cas9, it'll make a double strand break in the DNA. You can also add a cargo gene that it'll bring into contact with the DNA. And so when it makes that cut, then it'll insert the cargo gene. And so that's how you can add genes to the genome of your target. And so both of those, so the double strand break will be repaired with the cell's repair machinery, and usually there'll be an insertion or deletion. And then the cargo gene, the way that it's actually inserted and becomes part of the genome is through the cell's own DNA repair machinery. So I wanted to talk a little bit about that. There's many different pathways. These are just for double strand breaks. There's a bunch of other kinds of DNA damage that have their own pathways for repair. And so this is sort of a trimmed down version of some of the double-strand break repair pathways. And so there's a few patterns in this figure. So from left to right, it's the easiest with non-homologous end joining over here for the cells to perform, going to the more difficult. And that's because the least conditions have to be met for this to be possible, and then very specific conditions have to be met for this to be possible. So this is more difficult for the cell to do. This is much easier. And so that means that this happens more often. This doesn't happen quite as often. And then, so you'll also notice homology. So this requires no homology, so no repeated sequences or shared sequences of DNA for this to occur. Then just a small amount of overlap or microhomology here is required for alternative end joining. And then you have to have these direct repeats, so that amount of homology for single-strand annealing. And then you have to have entire sister chromatids for the homologous recombination. And so this will only happen in S and G2 phases where there's a second copy of the genome. And then the last pattern in this figure is NHEJ is the most error prone. And so it happens quick and easy, but it also happens with quite a few errors. And then this is difficult to do, and it's not very fast, but since it's using the sister chromatids as a template to repair the damaged, then this is very, very accurate. This has the least chance of error occurring. And so the title was through knockout of non-homologous end joining. So the particular proteins that I'm going to be focusing on knocking out are going to be Q80, which is one of the first binders after this pathway choice protein. But Q80 caps the ends of the double-strand break and then recruits more proteins. DNA PKCS is one of the later ones, and it'll phosphorylate chromatin. And then the very last in the chain is the DNA ligase 4, and so that's what actually ligates the DNA back together and finishes the repair. And so if we knock this out, then it should deny this pathway to the cells, and so it should send it towards these homology-dependent repair pathways, or the HDR pathways. And so the reason that we would want to do that is because single-strand annealing and homologous recombination are what's actually useful for gene drive and what's useful for making our gene drive quote-unquote biodegradable. So this is leveraged by gene drive. This is the actual mechanism that through which our version of gene drive occurs. And then this is what reverses the gene drive to make it biodegradable. And so focusing in on the non-homologous end joining proteins that I'm trying to knock out, Q80 is basket-shaped. It's called Q80 because it's 80 kilodaltons. There's a Q70 that is, would you believe it, 70 kilodaltons. I have no idea why it's called Q. That part of its name is a mystery. But it binds to the ends, and it can work with either short overhangs or just blunt DNA. And then DNA PKCS stands for DNA-dependent protein kinase, and it's just the catalytic subunit of DNA PK. And so that phosphorylates chromatins near double-strand breaks. And then ligase 4 cleverly ligates ends of double-strand breaks back together. And so just a review from yesterday of single-strand annealing. It can only occur when you have these direct repeat sequences flanking where the damage happens. And so direct repeat just means that these have the exact same sequence of DNA. And so if damage happens there, then these ends, just one strand on either end is resected by certain proteins. And so you have these single strand, you have these single strand overlaps. And so since the direct repeats have the same sequence and these ends were resected, then these are complementary, so they stick to each other. And so those stick to each other, but then you have these single strand overhangs. And so those are clipped off by other enzymes. and then it's ligated back together. So you've gotten rid of the double strand break, the DNA is repaired, but you've generated a fairly sizable deletion. And then homologous recombination, you need a sister chromatid. And so if damage happens and you have this double strand break, then the sister chromatid is used as a template, and then it's completely repaired, generally error-free. And so I'll go through gene drive again really quickly. So comparing gene drive to normal Mendelian inheritance. So if we release genetically modified males, we'll make sure that they're homozygous in the genetic modification. So both the copy from their mom and their dad will have the genetic modification. So when they mate with the wild type females, then you'll get these hybrid mosquitoes. And I have just males here, but obviously be male and female, but just simplifying the figure. And then if the hybrids mate with more wild type, then you'll only get 50% hybrid, and 50% wild type in the second generation. And then if that keeps going, 25%, 12%, 12.5%, and then so you can see that the genetically modified gene is not pushing through the population, it's being diluted in the population. And so that's why we want to do gene drive, and so that works off super Mendelian inheritance, so just greater than Mendelian inheritance. So we have a genetically modified male, mates with a wild female, and we get the hybrid again, but the difference with gene drive is this gene drive event happens, and it copies the genetically modified allele over the wild type allele. So you just get two genetically modified alleles in the same generation. So this is the same individual, and so it's just copied over. And so when that mates with a wild female again, then you're back to hybrid male instead of 50-50, but then the gene drive event happens, and so that just keeps going. And so this can push through a population and actually propagate and become established. And so super Mendelian inheritance is sort of a more broad term. So that's any time that a gene is pushing through a population. It could happen through natural processes. It could be just selective breeding. So it could be human initiated. Gene drive specifically is human initiated, but specifically using genetic tools like CRISPR or TALENs or homing endonucleases or something. But so that's the difference between gene drive and super Mendelian inheritance. Super Mendelian is more of a general term. So the way that the gene drive event happens is the homologous recombination. So we have this transgene that we've inserted, and this is the hybrid male that we're looking at. And so part of this transgene is something that it'll be some sort of nuclease that actually attacks the wild type version, or the wild type allele. And so it'll generate a double strand break in the wild type. And then if homologous recombination happens, then it uses the transgene as a template to repair the wild type. And so the transgene is copied over the wild type. And so that's how it doubles itself within that generation instead of, and so that it's passed on as homozygous instead of heterozygous. And so the way that we make that biodegradable so that we can test it without making any permanent changes to the ecosystem is something else that we'll add to the transgene is a different endonuclease that will attack itself. And we also make sure that the transgene that we add is bookended with these direct repeats. And so the transgene will code for a protein that will attack itself, and so it'll generate a double strand break within the transgene. And since we have these direct repeats, then single strand annealing happens. These ends are resected to make the single strand here. The direct repeats are complementary, so they stick to each other, and then those are clipped off, and all of the genetically modified parts that we've added are deleted. And so that's what makes it temporary. It makes it biodegradable. All right, so moving into the two different projects that I'm working on. The first one is an injection project, and so this is in living mosquitoes. And so the goal is to generate non-homologous end-joining knockouts of mosquitoes, and then see if we can push DNA repair towards the homology-dependent repair, or HDR. And then, so if that happens, if it's pushed more towards that, we want to quantify exactly how much more often HDR occurs than NHEJ. And so we do embryo microinjections. So this is one of our research assistants, Brian Contreras, and he's at our injecting microscope. And so you can see the embryos here in the screen, so that's what he's seeing through the eyepieces. And then you can just barely see the needle. And then we keep the embryos under oil to keep them from getting dried out. And so you can even see the injection fluid with the Cas9 because the injection fluid is aqueous, so they don't mix. And so you can see this little bubble here right next to the embryos is the injection fluid, and then the little line is the needle. And so we're injecting these embryos. And since we want them to pass on these genes, we want to make sure that if none of their other cells are affected by the Cas9, at least their gametes are affected. And so we inject in the posterior end because that's where their gametes are developing. And so we have to line them up, and we can't just line them up anyway. We have to line them up with the narrow end facing all the same direction. And so they're similar to chicken eggs, where one end is more pointy than the other, but it's not quite as obvious as chicken eggs. And so you can see some of them are more obvious that they're kind of pointy, but then some of them just look like perfect ovals, and you have no idea. You just kind of have to guess. But we inject them one at a time, just down the line. And then what we're injecting with is Cas9 complex with the sgRNA that's targeted to a specific gene called KMO, which stands for kynurenine 3 monoxygenase, or monooxygenase, which is a enzyme that just, it's in the pathway of making their eye pigment. And so, so we use that as an insertion target and a reporter. And so I promised that I'd explain why they have different colors, so this is why. And so the sgRNA is targeted to exon 4 of the KMO gene. And so looking closer, this one is normal. That's what the eyes normally look like. So that's full eye pigmentation. And then this is what we call mosaic eye. And so the reason that it's mosaic is because this is, when this pupa was an embryo, it's one of the ones that we actually injected. And so the Cas9 doesn't get to all of the cells equally, and so it won't necessarily cause the double strand break in every single copy of the gene in every cell. And so the ones that have the dark stripes, those came from cells, from probably stem cells, that the Cas9 didn't quite get to, so they're still producing pigment. The white ones here, the Cas9 did get to, and so that gene is knocked out, so they're not producing pigment. And so the exon 4 is the target, and then the reporter, we can look at their eyes as a reporter. And so if they have white eyes, we know that that gene's knocked out, or we've done something to that gene, so that lets us know that our Cas9 is working, our sgRNA is targeted to the right place. But we can also insert a gene right in the middle of that KMO gene. And so since it's inserted in the middle of the KMO gene, when we insert GFP, it knocks out KMO. And so when they're homozygous for this GFP modification, they produce GFP throughout their body. So that's based on the promoter we use. We use a promoter that expresses it ubiquitously in any cell type, so it's expressed in all tissues. We also have other promoters that usually we use it with red fluorescent protein that will only express in the eye, so it makes their eyes red, like glowy red, and it's pretty cool. I don't work with those, so I didn't get any pictures, but I realized that I should have talked to some of my friends and gotten some pictures because I know y'all want to see it. So just taking a closer look, since the GFP is inserted in the middle, it's knockout. So if we look at this one, you can see that the larva has white eye compared to this one that is not expressing GFP, you can see so this is what black eye looks like, and so you can see that it's white eye. That one might be, no, I don't think that's something else. That's not an eye. But anyway, so that's what is going on there. And so for my experiment, what we need to do is cross these GFP expressing ones with some of the non-homologous end-joining knockout ones. And what we need is them to be heterozygous with GFP and wild type. And so, and I'll explain exactly why we want heterozygous instead of homozygous later. But all right, so in the NHEJ proteins, we have different mutated alleles. And so each of the different genes has their own challenges. And so if we get full knockout females with Q80, they're not fertile. And so when we're doing our genotyping, we'll get knockout males and then cross them with heterozygous females, which means that only 50% of the embryos that we inject are going to be full knockout, it means there's an extra genotyping step so that we know whether we injected a full knockout or a heterozygous embryo. But it also means that we get more data because not only can we compare knockout to the control, but we compare full knockout to partial knockout to the control. So turns and roundabouts. So what we really want is what's known as a transheterozygote. So heterozygote means there's a mutant and then the wild type. Homozygote means it's the same mutant for both alleles. Transheterozygote means it's mutant for both alleles, but it's a different mutant. So the ideal is getting the transheterozygote because this is full knockout and they're different. So it's just more likely that the cells won't find a way to get around that. But the homozygous knockouts are also okay. We just prefer these. And so just to simplify the notation, we'll just use minus minus to show that it's full knockout, but not necessarily say which kind of knockout. And then similar with DNA PKCS, these on paper look like the most straightforward. The full knockout females are fertile. It's not sex-linked. It should just work, but I'll talk a little bit later why it didn't just work. But we have different mutants, and so the transheterozygous, again, is ideal, but the homozygous knockouts are okay, but I'll talk about that later, why that's not quite okay. But just simplifying the notation, the minus minus for the full knockout. And then ligase 4 is sex-linked, and so we have 3 mutants. Capital M is analogous to like the Y chromosome. Lowercase m is analogous to like an X chromosome. And so to get the transheterozygous, so we can have the male delta 11 with either delta 2 or delta 7, and it's a transheterozygote. And then the reason that we needed a third one is so that if we get females, we can still have transheterozygote and females. So, and then females, we can also get homozygotes, and those are okay, just not ideal, but we can still use those. And so just simplifying all that to the minus minus notation again. All right, so the way that we do genotyping is a way that's a little faster and easier and a little bit cheaper than just full sequencing. So it's called high resolution melt curve analysis, or just HRMA. And so, yes, genotyping sequence or without sequencing. And so it works off this principle that different base pairs will melt at different temperatures. So if you have CG pairs, they have 3 hydrogen bonds, so they melt at a higher temperature than the AT pairs, which just have 2 hydrogen bonds. And so based on the sequence, the melt curves will be different. And so there's also a special dye that only fluoresces when it's bound to double-stranded DNA. And so when you have an intact strand, the dye is attached to double-stranded DNA and it's fluorescing. And then as the temperature increases, it melts, and so they come apart based on the sequence and the number of hydrogen bonds, so they come apart, and as it comes apart, the dye no longer fluoresces if it's not bound to double-stranded DNA. And so as the temperature increases, then the fluorescence will decrease based on the sequence. And so you can see the red and the green, they melt faster, and then the orange melts a little bit later, and then blue, and then gray. And so we know which of these should be our wild type controls, and so we'll normalize these curves to the gray ones that we choose as our wild type. And so we'll get something like this. And so these gray, that's just straight across here, is our wild type. And so then our different mutated alleles have different curves. And we've done these a lot, and it's the same mutated alleles, and then we've done actual sequencing so that we know exactly what we have. And so we just have a cheat sheet that has the shape of these curves that we can just compare to. And so this one, this green double peak, we know that that's a delta 8 heterozygote. And then the orange is the delta 1 heterozygote. The red is what we really want, which is the trans heterozygotes. And then these are very similar curves, and as far as our experiments go, these are basically in the same category, so we don't even distinguish between the two different types of heterozygotes. But anyway, we can tell those apart. And then DNA PKCS, similar. This is the most recent data that I have. And so this is, I mentioned that it should have been the easiest on paper, but it wasn't. And so we're doing genotyping, and they're not sex-linked. They're not, the females are fertile even if they're full knockout. So it should have been easy, but we just could not find the trans heterozygotes. Most of what we got was something that looked like this. And so we'd cross a heterozygote with the other heterozygote, and we'd expect at least 25% to be the trans heterozygote. Nothing. It was just delta 38 group and a delta 10 group all the time. And so I took a closer look, and I realized that some of these are a little bit different shape. So some of them come down and they dip a little bit lower and they're more curvy. And then some of them are more of a straight shot down. And so I went in by hand and just changed the color, and I was like, I think the trans heterozygotes have been hiding this whole time behind the delta 38s. And so I did sequencing on some of these to see exactly what we got, and the trans heterozygotes have been hiding behind the delta 38 heterozygotes the whole time. So I haven't gone back to the months and months of failure to check and do sequencing. I'm not sure if I have the emotional fortitude to do that, because I may have just wasted about 4 months of my life. But anyway, we know where the trans heterozygotes are now. All right, so getting into the actual experiment. So NHEGA positive, that's just our wild type controls. So these are non-homologous end-joining competent mosquitoes, and so we haven't messed with any of those, so that's just wild type. And so that's one gene. The other gene is the KMO gene. And so we want heterozygous for GFP and KMO. And then we have all of our knockouts. And so this gene's knocked out, and then heterozygous in GFP, knocked out, heterozygous in GFP, et cetera. And so the reason that we want it heterozygous in KMO is because our Cas9 with the guide RNA is targeted to the wild type. And so it'll only cut the wild type, so it'll generate a double strand break here, and then one of three things will happen. Either the Cas9 didn't quite make it, so we'll get no damage, or the cell will get very lucky, and we will get very unlucky, and non-homologous end joining will actually have errorless repair, or something crazy will happen. It's possible, it's just not likely. And so we'll still have functional KMO gene. Or HDR will happen, and so we'll have homologous recombination. The GFP will get copied over, and so this will be basically that gene drive event, so it'll just double our alleles of GFP. Or we'll have NHEJ happen. There will be some sort of insertion or deletion when it repairs it inaccurately, and so we'll have KMO indel, which is basically knockout. It's just not necessarily the knockout that we have as an established line. And so these are the G1 mosquitoes. And so we raise them to adults, and so we'll have, so the GFP heterozygote, so that's this one again, and then the GFP homozygote, so that's if this happens, and then the transheterozygote with GFP and knockout, so that's this. And so we'll cross that with a line that we have that it's just an established line that we keep going, but it's full KMO knockout. And so the offspring will be one of three things. It'll grab this allele, and it'll be positive negative, or it'll grab any of these 4 GFP alleles, and so it'll be G negative, or it'll grab this knockout, and it'll be full knockout without GFP. And so what that looks like, and the way that we actually evaluate our data, is we'll look at the mosquitoes, and so if no damage happened, or if we just got unlucky and there's errorless repair, then we'll have basically just normal mosquitoes. They'll be black eye, they won't glow green or anything. 50%, or up to 50% of them that glow green will be just what we expect, because 50% of the alleles of all the G0s are going to have GFP. So we expect up to 50% GFP. And then anything beyond GFP is, so let me go back. So so there's six alleles here, 6 possibilities. And so 3 of those are GFP, so half. And so anything beyond half is going to be this 4th G right there. And so anything beyond half, that's indicating that HDR was happening. And then the last possibility is a mosquito that's not glowing, but its eyes are white. And so we have full knockout of KMO, but there's no GFP. And so that indicates that NHEJ happened. And so we had some error in the repair here, but no copying of the GFP. All right, so moving on to the primary cell project. So this is taking all of our lines that are knockout and then trying to just dissect them and get primary cells to culture so that we can do microscopy on cells and do antibody staining or staining for live cells and just looking at them at a molecular level. And so this is to connect the macroscopic, looking at living mosquitoes, to the microscopic, so what's actually happening within the nucleus, which is where all the DNA is, and seeing if what we're doing with knocking out NHEJ is changing the nuclear architecture. And so this is much similar to what I did in grad school and not in my postdoc. So I'm just trying to connect those two. And so the first thing that we need to do is collect tissue. And so we can either crush tissue, and so that's mostly useful with embryos. And so you basically just get eggs and just crush them with tweezers. It's not very high throughput, and it doesn't work very well. You can homogenize, and so that's basically a mortar and pestle. You can either do that with eggs or larvae. And so the mortar is basically just an Eppendorf tube, and then the pestle is just a tiny little plastic pestle. You just, that is kind of messy, and it doesn't work very well. You could also vortex, which is pretty fun. You put beads in a centrifuge tube, and then larvae, and they're all swimming around happy, and then you put them in the vortexer, and it just annihilates them. It's hilarious, but not very useful. They get all over the beads, and they get stuck to the beads, and then you have to strain it. It's a mess. And then we can just eviscerate the larva, so that's with a microscope and tweezers. And so we have some nice larvae here, and then we have some former larvae here. So I'll talk more about this. It didn't work great either, but I'll talk more about it. And then the last thing is dissection. And so we do that mostly with adults. So that's dissecting out their midguts. Going to the larva for the evisceration or vortexing. So we incubate the live larva in like a 6-well plate with just water, and then we'll add these antibiotics and then this antifungal. And so that's so that as they're eating and drinking and stuff, they'll be taking in antibiotics and antifungal, and that should clean out their guts. And then we wash the outside just before we process them. And so we have this cereal wash with ethanol, bleach, water, and that kills them, but that's just so they don't have to feel being ripped apart with tweezers. And so that should wash the outside and get rid of all the bacteria and fungus and whatever on the outside. Because larvae are just in their pans in water, living in their own filth, and so we don't want that, because cell culture is very clean. If you're culturing cells outside of a body that has an immune system, then you have to provide their immune system. And so just keeping everything clean, culturing stuff with antibiotics, is you giving the cells their immune system back. But antibiotics in solution can be overwhelmed, so you want to clean the larva first. And so one of the problems with the larva is one of the mosquitoes' immune responses. And so this is pretty common across all insects, is the melanization response. And so it's basically like if you get a cut and you have a scab, then what the scab is basically like this protein sheath protecting the wound. And so the melanization is basically just this sheath that they lay down around either foreign invaders, so they'll melanize bacteria, or they'll melanize it over wounds. And so if we're ripping them up with tweezers, then their entire body is a wound. And so these are some eviscerated larvae very soon after I had eviscerated them. But if you just let it sit, then this phenyloxidase, which is the enzyme that catalyzes the melanization, will just melanize everything. And so the whole thing turns black. And so microscopy works by passing light through. And so if everything's just black, then it's not great for microscopy. And so one of the ways that we tried to get around this was using kojic acid as an inhibitor. So kojic acid inhibits phenyloxidase. And it sort of worked. We didn't really get any melanization. But we're already adding extra antibiotics, extra antifungal, now extra kojic acid. And the other problem is that eviscerating larvae is just not high throughput. It takes an hour to eviscerate like 50 larvae, and you need a lot more larvae than that to really get enough cells from them to culture. And so the protocol is just becoming more complicated and not really worth it. So I moved on to midgut dissection. And so we'll feed adult mosquitoes their normal meal is sucrose, 10% sucrose, but we'll add 1% pinstrap. And we'll let them feed on that for 1 to 2 days. And so that'll clear out their gut. And then we dissect their gut. And so we get to rip off their legs and their head. I mean, we have to rip off their legs and their head. And then we'll pull off the last 2 segments of their abdomen. And all these tubes are connected, so it just kind of pulls the midgut out through the bottom of their abdomen. And then we can pull off the extra tubes and everything. And then we just have this midgut. And we can drop 1 to 5 midguts in a well, and that'll be enough cells to pretty much cover the well. And so since we don't have to do as many of these, this is much more high throughput. It's just a lot easier. We discovered that we still have to add some extra antibiotic and extra, antifungal to the wells that we wouldn't do with normal cell culture. So we're still adding a little bit, but it's not nearly as complicated as doing the larva was. So this is what we're going with most recently. And then something else, the midguts generally kind of just fall apart and their cells just kind of leak out and go do their own thing on their own. But I was finding that took a little longer than I wanted. And so after about 2 days, I just go in there with a pipetter and just pipette them up and down. And that's enough mechanical stress on the midgut to really help them break up and just expel many more cells. And so the main challenge now is we're getting a lot of cells, we're getting a lot of separation, they're all spreading out. They're just not adhering to the plastic, which most cell lines that are immortalized that you work with, they adhere to the plastic, so they spread out, which is a lot more useful for microscopy because when they spread out, they get thinner, so you can see more in the microscope. And then also when you change media, if they're adhered to the plastic, the cells stay and all the debris goes. But if they're not adhered, then you'll just get rid of cells and debris. So it's not as useful. So trying to get them to adhere to the plastic is the next step, but we're at least getting plenty of cells to work with. And so this is just some of the most recent pictures that I've taken. Host is a stain that specifically stains DNA. So anything blue here is DNA. Specifically, the little points are nuclei, and then the lighter blue area around them is just kind of autofluorescence in tissue. So this is many cells together. Maybe some of the smaller dots here are individual cells, but mostly this is still kind of a chunk of midgut that hasn't quite fallen apart yet. But anyway, that is basically where that project is. And how am I doing on time? Was that another hour and 7 minutes? My practice was an hour and 7 minutes long, so hopefully I went faster than that. We have time for questions? Okay. So Brian Contreras, that was my wonderful model at the injector. And then our principal investigator is Dr. Zachary Edelman, so yeah. Yes?
When you were designing the guide RNA, did you have any trouble finding a PAM site that was in an area that you could use to knock out?
So he is asking about designing the guide RNA if we had difficulty finding the PAM site? Is that?
Yeah.
All right, so I have a squirrely answer for that. I am just using guide RNA that was already used in our lab, so I'm not the one that designed the guide RNA, so I don't know the answer. Sorry. Yes.
On whenever you were going over like how gene drive works, how do you govern the timing of when your transgene lyses the wild type and then whenever it cuts itself out?
So that is what we're doing research on, is trying to tune those knobs. So the gene drive, I think, so for the actual gene drive, that relates more to my work. And so knocking out the NHEJ should make that happen more often so that the homologous recombination happens more often. And then the tuning the single strand annealing to cut itself and then delete it, that's one of our PhD students. And so what he's doing is he's looking at different homing endonucleases because they'll have different efficiencies. And then he's also looking at different target sites and looking at whether it does it more often if we have multiple target sites or if we have like multiple homing endonucleases that are attacking multiple sites, or I think he's also looking at the length of the direct repeats, and then also the spacing in between the direct repeats, because the further away the direct repeats go, the less likely it is that you'll actually have SSA, because the repair machinery won't be able to recognize the direct repeats. And so he's looking at all that to kind of tune those knobs. And so one of the papers that we've published, it was, I think, just last year, or it might have been in 2020, but it's the one titled Making Gene Drive Biodegradable. So we've done, or previous people in the lab have done the computer modeling to see how often that SSA event, where it attacks itself, needs to happen for the gene drive to be able to push through the population, but also delete itself. And so we know how efficient those endonucleases should be. And so it needs to happen about 10% of the time per generation. And so what we're working on now is tuning that so that we can actually make it 10% of the time. And so, yeah.
infested mosquito and you got it right, do you have a government who's gonna let you send it out into the environment? Is there somebody who wants this? Like, how is this gonna actually look? And then will it be like letting a few fly off? And like, what does it look like if this really comes down to
Yeah.
pass?
So one of the main things is working with local governments or state or federal governments, and just sort of like when you do human trials, you have to get informed consent, and so it's basically getting like community informed consent, and so explaining what we're doing, what the risks, what the benefits are, and then talking to governments, but also just like community leaders and just people that live where we're going to do this. So that's sort of the preparation, and then there's been releases, at least of sterile males, but maybe gene drive. I'm not sure about gene drives from other labs, but there's been releases. I think they do sterile males in like California and Florida, and there's been some in Brazil, but countries where malaria is a huge problem or dengue is a huge problem, they're much more willing, like if you've done your homework and you have good science and they can see that you know what you're doing, then yeah, they don't want 40,000 people a year dying of dengue, or over 500,000 people dying a year of malaria. So as long as you can show that it's safe, then there's definitely governments that will want you to do that. And then the biodegradable is what we're trying to develop, that because that lets us get more data about whether a gene drive is safe, because we can do the experiment in the wild and it's not permanent. And so if it deletes itself, then we can do a temporary experiment in the wild and get data in a real population instead of just trying to get a giant cage, and that's not very realistic to the wild, and just have a huge colony of mosquitoes and see if it pushes through that population. That's useful. It's just not as useful as if you could do it at scale but temporary. So, yes, I think Allie, yeah.
Continuing with Ms. Rogers' question too, are we talking about the release of 50,000 genetically modified organisms at a location? And what is the mosquito's range? Like, do they stay within a 50-mile range, or is this a 5-mile range, or I don't know. I'm going to have to go read the books, but I'll maybe.
Yeah. I'm gonna have to go read the books too. I'm new to entomology, so I don't know what their range is. And then our lab is still pretty far from an actual release, and so I'm not sure of how large these releases would be either. But so.
I heard there was a guy with his own private island, and he was willing to let you try this out there first, somewhere near Edinburgh.
Oh.
Some kind of park.
Oh.
He was
All right.
very amenable to you.
Yeah. That was in my other talk, the Jurassic Park. Yeah. John Hammond's kind of a loon, but.
I had a question similar to Dr. Laughlin.
Yeah.
In terms of the lifespan of a mosquito.
Oh, yeah.
The range of the mosquito, the average reproductive cycle.
Mm-hmm.
How far conceivably could your temporary experiment reach?
So the life cycle of the mosquito, once the eggs have been laid, the embryos are typically developed enough to hatch after like 3 to 5 days, and then they stay larvae for about a week and a half, and then they're pupa for maybe 2 days plus or minus, and then they emerge as adults, and then the adults will live for 6 weeks, more or less, and so their whole life cycle is maybe 2 months, but they're sexually mature by like 2 weeks. They're usually able to start mating after a day or two from emerging,
So,
and yeah.
You said the males would mate for a time and then move on after a resting period. So conceivably,
Yeah.
one of your sample males could prolifically affect an
Right,
area of
yeah.
some extent for about a month.
Yeah, so the males don't live quite as long, but they can mate 1 or 2 days after they emerge, and then they can mate with 3 to 5 females. And then after about 3 or 4 days of rest, they can mate with 3 to 5 more females. And so, and they'll live for maybe 4 or 5 weeks. And so, yeah, they can spread the gene pretty far, just a single male. So, yes.
So, you've had a lot of experiences here and beyond here. What do you think prepared you best for the research you're doing now?
For the research I'm doing now,
I know we have a lot of students in the audience
Mhm.
kind of looking forward to what they're going to do next. So what kind of things did you get to do, opportunities you got to have
yeah.
that have helped you with this right now, with your job now?
So I'm definitely using genetics more than I did in grad school. So Dr. Martin's genetics class. And then the genotyping and stuff is a lot of like molecular biology lab work. So cell biology, analytical biotechnology as far as classes go, and then like the FDA internship that I did one summer. I did a lot of lab work there that helped me practice. I think I helped Josh with like E. coli stuff, like maintaining colonies and stuff. I helped him with that. Yeah, just lab experience like that helped. And then rearing mosquitoes. I oversaw some rush parties. Like you know, I was like, get busy batch. So. Yes.
Obscure movie reference. So we had Jurassic Park, and today we had Gattaca. What's next?
What's next? Hopefully not I Am Legend. Yeah. Or Resident Evil. Yes.
Speaking to that, what is a potential danger with this kind of research?
So for mine knocking out their DNA damage repair, if we're trying to gene drive that or something into a population, that's a liability. So there's minimal risk there because those will just eliminate themselves. There'll be a selection pressure against knocking out DNA repair proteins. If we did something else with the mosquitoes, or if we just eliminated Aedes aegypti entirely, we don't know of any reason or any. Most of the jobs that mosquitoes perform in the ecosystem are redundant with other insects. So we don't know of anything specifically that could be a risk if we just eliminated populations of mosquitoes that spread disease. But we also don't know what we don't know. And so we could eliminate that, and then that causes some worse problem. And so that's one of the reasons that we want to make it biodegradable, so that we can test that, and it's a temporary thing where we knock the population way down or we eliminate it, and then we see, like, is there any problem? And then, so we have actual data, we're not just guessing, so, yeah. So.
I know we're almost up with time, but I do have maybe one last question. Are you in competition with others? Is there a race going on?
I guess sort of. Like there's a lot of labs that are doing gene drive, and everybody kind of has their own method. They're not all the exact same way of doing gene drive, but they also have different things that they're trying to change with the mosquitoes. I'm not sure if anybody else is trying to make it biodegradable. There may be, but I'm just not, I don't know of anybody that are, but I'm also not familiar enough with the literature to say that there definitely are not, so. So there's definitely a competition with the gene drive. A lot of people are trying to be the first. Our PI is very meticulous about getting things done right, and so we're not necessarily trying to be the first. We're trying to make sure that what we have is rock solid. So, which I think is why we're focusing on the biodegradable part of it. Yeah. Oh yes, was that? Are you just okay? Just stretching. All right. Yep.
I'll ask the last one too, and probably stop.
Okay.
I get one question. Okay, so I always assume nature's smarter than we are. Right? Nature wins.
Mm-hmm.
Are there known or potential mechanisms where the population, do you expect to have individuals that would be resistant to the gene drive? Are there mechanisms for that?
The gene drive itself, since there's so many ways that damage could be repaired, the gene drive itself is kind of difficult to do because they have all these other options of ways to repair that damage so that it's not copied over. Actually the elimination of it also is just going to be inefficient because just because there are direct repeats doesn't necessarily mean that it's going to go down the single-strand annealing pathway. But that's also, we want that to be somewhat inefficient just so that the gene drive can happen. Is there anything that would
So,
specifically, yeah.
My question is, if you release it in the wild, do you really expect it to be 100%, or is it somewhere between, you know what I'm saying?
Right. Would it completely eliminate itself? Nature is complicated, so there's always a chance that it's not 100%. But is there anything specific that would cause that? I think just the inefficiency of it going down that particular pathway in general would make it more difficult. But because NHEJ is always going to be easier for the mosquitoes to do. So.
No, obviously nobody's trying to do that.
Yeah.
All right, please thank Daniel.